Okay, so it's 4 o'clock here. So I'm going to get started. Welcome, everyone, and shalom to our seminar in our Australia-Israel Scholar Seminar Series. I'd like to start by acknowledging the Turrbal and Yagara people as the traditional owners and their custodianship on the lands on which we meet today. I pay my respect to their ancestors and their descendants. And I extend that respect to those joining us from around Australia and in other Indigenous lands around the world. Today, I'd like to welcome our speaker, Professor Nirit Bernstein from the Volcani Institute in Israel. And her talk today will be entitled Advances in Mineral Nutrition of Medical Cannabid Plants. Professor Nirit Bernstein is a principal research scientist and head of the Cannabis Physiology and Agronomy Lab at the Volcani Institute of Agricultural Research in Israel. And her research focuses on cannabis plant science and development of cultivation techniques for cannabis sativa. She has her PhD from the University of California, Davis in the US, and a Bachelor of Science from the Hebrew University of Jerusalem in Israel. So I'd like to welcome Nirit Bernstein to begin her talk. Thank you.
Hi, thanks for inviting me and thanks for having this platform. It is very nice to have in such a not easy time. Anyways, I'm going to show you some of our research that we've done in the last couple of years in medical cannabis. So first of all, we have to say that cannabis is not a new crop. It's one of the most ancient crops known for humanity and used by humanity. And it was used mainly for medical reasons. Because it has a medical value, obviously, it was also considered to be involved in a religion or traditional beliefs. It has a very large value. It's a fiber crop, so it's an industrial crop for production of fibers and ropes and construction material and so on. And always throughout the years, recreation was actually elevated greatly during the last decade. And let me go. And normally, I mean, this is not only ancient history. I mean, cannabis has been known to humanity for thousands of years and a couple thousand BC. But also, if we were to open a medical cabinet in the last 150 years or so in Europe, in the US, whatever in the world, there would be a lot of cannabis products. So this was one of the medical powers and advances that humanity had. It was considered a very successful medication. And let's not forget that even up today, more than about 85% of all our modern conventional medicines comes from plant materials. So it's nothing is unique about the fact that the plant was considered to be a very strong, powerful drug to be used for medicine for a long time. So if everything's so great, what happened? So what happened was in 1961, with initiatives that started in the US for reasons of worthy of maybe another seminar, but basically convinced most of the world, developing countries and developed countries, to sign an international single convention on narcotic drugs, that cannabis was put in it as among one of the most dangerous drugs. And in that point of time, cannabis was defined as a narcotic substances, which is not registered and not allowed to be used as anything, definitely not as a medical product. And this convention basically stopped all research and all medical use because for 1961, no research on cannabis. But this treaty says basically no cannabis. You're not allowed to grow cannabis, not to buy cannabis, not to sell cannabis, not to import, not to export. And two surprising thing is not to use it for medicine and not to use it for research. But it also say unless the specific country will have regulation on how to do it. The world will basically stop from 1961 until 2016, which is when Israel was the first country to set up regulation on how to do it. So the government of Israel, because the treaty says that if there will be a government government unit and rules on how to use it for medical and how to use it for medicine, then it is allowed based on the International Convention Treaty. So this is what happened in Israel. In 2011, there was a series of government resolutions. 2011, the Israeli medical cannabis was established. 2013, it was ruled that we need to improve knowledge and research in order to be able to give instructions on how to use it for medicine. And in 2016, which is a memorable year for us, what we call the medicalization model was approved, which was, again, the regulation on how to use it. And the cannabis was basically open for research, for medical use on a more wider basis. Of course, everything is highly regulated and tied. Every patient needs to go through a very rigorous process of testing for family history, medical history, to make sure that, the negative aspect of cannabis will not affect the family. At the same time, something also happened in the same year, 2016. The Ministry of Agriculture in Israel have declared cannabis as an agricultural crop. It's a legal definition that says that from this point of time, we are now able to apply for funding to do research on agronomic from agronomic side on how to use it. So something very important to understand about cannabis, that it's unlike other medications that most of them are produced today in factories. We're talking with a plant material, and the usage for medicine is based on the plant material or extract from the plant material, meaning that the way that we grow the plant has an immense effect on the product. And so it has to be an integrated process between researchers on the cannabis agronomy side, the researchers on the medical side, to make sure that we produce the product that's actually needed. So at this point of time, all of a sudden we found out we are first in the world that are allowed to do legal research on this plant. And with the changes in regulation, cannabis is gaining a status of medical or recreational product in other countries. But what do we know about how to grow it in modern cultivation? Research stopped in 1961. Think about the advances that happened since there. Greenhouse cultivation, environmental control, analytical chemistry. We are stuck in history. And all of a sudden, there is a huge need to close the knowledge gap and to develop this into a modern cultivation crop. So there is a need to develop innovative agro-cultivation technology in order to support this booming cannabis industry. About 11 years ago, at the time I was working on the effect of salinity stress on plants and cell division, cell elongation, and fertilization, and whatever in remote cases in Israel, and high intensity on crops. I received a call and they say, Dr. Bernstein. I said, yes, Nirit, how can I help you with it? And they say, our growers have problems, the leaves are not developing properly, we need really a lot of help on the scientific side. Are you willing to help? Well, I'm not an extension service scientist, but I work for the Ministry of Agriculture and Botany Center. I said, of course, which crop is it? And they say cannabis. And I tell myself, oh my God, drugs, right? Well, this happened to be the Ministry of Health in Israel that decided that in order that there's a need, first time in the world, to regulate cultivation, to make sure that what we give our patients is regulated and safe. So this has been ever since, that we've been covering in my lab a huge range of topics because the level of knowledge was very low. And there was a huge amount of challenges. We also make a considerable number of breakthroughs, which are now used in Israel and worldwide. And for example, we need to optimize yield quantity and the chemical quality, which is the therapeutic quality. And we need to standardize this to make sure that what's in the top of the plant is the same as in the bottom of the plant, and what we cut in January or December is the same, because it eventually all goes into a bag and given to the patients. And if there will be 2-3 times difference in the concentration of compounds, then patients will be in trouble. So we've been checking effect of mineral nutrition and light quality and light intensity and temperature and pruning, plant architecture, a lot of things on the product. How do we store this product post-harvest, after we cut it or before it's packed for the patient? How do we prevent contamination, microbial, heavy metal, pesticide and insecticide? Pest management in general, which we were working on developing botanical pesticide instead of conventional chemicals which are not toxic. Flowering got a huge number of topics, and today we will discuss mainly effect of mineral nutrition. That in a minute I'm hoping to convince you in a few minutes that it's a key issue in quality and development of the plant. So how can we affect plant performance and to get from the plant what we need for our patients, which is a safe and standardized product? So first of all, on cannabis, one thing that we need to understand that unlike most medications that usually will have one compound, we're taking antibiotics or whatever, it's one or two compounds. Cannabis have what we know today about 1,000 compounds which are biologically active, which means they can affect our body. And these are divided mainly into three groups. Cannabinoids, we now know close to 200 cannabinoids, not only the THC and CBD that people are talking, more familiar with. Hundreds, terpenes and flavonoids, which are the same as what we found in other plants, which are responsible to the smell of the plant, but together they have an effect on the plant. And all those compounds are affected by the production in the plant is affected by two main things. One thing is genetics. Different strains of cannabis will have different chemicals. Not every plant have 1,000 compounds. It's the same thing that if we look at tomatoes, and we have large tomatoes, cherry tomatoes. Today we have purple tomatoes, whatever. Each one of them is different. In cannabis, the same thing. So the cultivar has a large effect on the potential of what this plant can produce in terms of chemical, therapeutic, or biologically active compounds, secondary metabolite, we call them. Second most important thing is how we grow them. The fact that there's a potential doesn't mean that the plant can fulfill this potential unless we actually give it this certain condition. And there's a huge sensitivity of the plants to environmental condition, and this greatly affects the secondary metabolites, which are the cannabinoids and the terpenes, which are produced by the plant. So what we know today, there's a huge challenge because we know so little today. Research have really only started. We're closing the research gap, the knowledge gap as soon as we can, but we definitely know it's only the tip of the iceberg. So cannabis is a magical plant, right? It's impossible not to say it. Let's stick to the medical potential instead in terms of medical potential. And when we look at the first, like 11 years ago, when we knew very little about cannabis, and we look at the range of medical indication that cannabis had, we said, well, it seems like a snake juice, right? It affects cognitive and neurological and skin condition. Well, how can this be? This can really be because we now know that cannabis has more than 1,000 compounds with biological activity that can affect our body. It's basically an entire medicine cabinet. So that's why it can affect. And now our role as an agronomist is to focus and to make sure that on the medical side, they need to know which compounds are required for whatever medical indication, and that's where research is going. On the agronomical side, we need to know how to produce the plant that contains standardized level of what's needed for medicine. So talking about mineral nutrition, we found out a huge sensitivity to many of the nutrients or fertilizers, and I would like to show you some of our results. So first thing that we did, cannabis is what we call the short day plant. It has a vegetative state in which it will just grow leaves and roots and stems and will not flower. And then when you get an induction to flower, it will start flowering. So we have to study requirement of the plant and behavior both on the vegetative growth phase and when it's flowering on the reproductive, on the flowering stage. So for example, this is an experiment, one of the first experiments that we did on the vegetative growth phase. We asked the question of how much potassium does this plant need? How much do we give it in order to get what we want from the plant? So we took a huge range of potassium from 15 milligram milliliter, 15 ppm to 240. And obviously, plants are very small and deficient and hungry. And it's obvious even by eye that somewhere between 100 and 175, this is the right range. Of course, this is all backed up by morphological studies and physiological studies and chemical studies, and this can all be found in our publication, which are usually listed here below. So this is what happened, but most importantly for the reproductive stage. So this is when the cannabis is growing. Again, causing it from 15 all the way up to 240. This is crazy. In agriculture, we usually use around 100, 150. And indeed, if we look by eye, we see this is deficiency plant. They suffer the high concentration. There's already toxicity. So probably somewhere around this range, around 100 ppm, gave us the best performance. This is, of course, measured many, many parameters in biochemical, physiological, as a function, but it's easier to look at the photograph at this point and in the time restriction. But what happened when we checked which plant is actually producing the largest amount of the cannabinoids and terpenes, the medical products? And to our great surprise, the highest concentration actually appeared in those very hungry, deficient plants. So this is for potassium. What happened for nitrogen? So, okay, nitrogen, phosphorus, and potassium are the three macronutrients that the plant takes up in the largest concentration. We did the same thing for nitrogen. We checked from very low concentration, but were obviously expected to be deficiency, all the way up to concentration that can be toxic. This is at the vegetative stage. And just by looking at the size of the plants without looking at all the figures that also give us the same, this is, for example, biomass of the leaves and biomass of the root, it's obvious that around 160 milligram per liter, this is what we want to give the plant. Many of the growers at the time were given above 320, which means they're wasting a lot of money, they're contaminating the environment, and they're harming the plants. So really, we're trying to supply the research-based information to the growers, and some of the growers, so we have some traditional growers that have a lot of knowledge, obviously, but it's now starting to be backed up by science. And what happened again when we looked at the medical qualities of the concentration of cannabinoids and terpenes? And whenever I'm saying medical, it can also be obviously recreational and life quality, because these are the same compound, cannabinoids and terpenes. And again, under 30 ppm, incredibly small, hungry plants. 320 ppm already starting to see deficiency sign. We'll probably go for 160, judging by plant function. When we checked again the concentration, the best concentration, highest concentration of those cannabinoids and terpenes appeared in the stress plants, in the deficient plants. We also checked for different type of nitrogen supply. So the plant can take up nitrogen as ammonium or as nitrate. So we gave 0%, we gave everything as nitrate, meaning 0% ammonia, all the way up to 100% ammonium, meaning 0% nitrate. And we found out that the best plant performance was only under ammonium, only under 100% nitrate nutrition, and also the highest concentration of cannabinoids and terpenes. Some examples here, the percentage of ammonium and the highest concentration of most of the cannabinoids. We have a lot more in our publication. We took dozens, of terpenes and cannabinoids. This can be found in the publication. So less ammonium is better, but we still get, even all the way up to 50%, we get reasonable levels of those compounds. What happened for phosphorus? So we've seen nitrogen and phosphorus under hungry deficient plant, highest supply. What happened with phosphorus? Basically, the same thing happened with phosphorus. People were saying at the time, cannabis is hungry for phosphorus. Give it huge amount of phosphorus. We said some growers were saying go 300 ppm, go 1,000 ppm. This is crazy in terms of agricultural convention. We said, wow, this may be a plant that needs a Nobel Prize if we actually found out that it requires so much phosphorus. And what we found out that I tried an experiment from 5 ppm, which is very low, all the way up to 90, which is for agriculture is very high. And we found out that in terms of plant development, best plant development occurred under 30 ppm, which is conventional agricultural, on the highest side of conventional agricultural. But again, when we check in terms of production of secondary metabolites, highest concentration were in the hungry plants, in the deficient plants for phosphorus. And this can be seen, for example, if we look at the concentration, we look at some of the cannabinoids that we tested, highest concentration usually appeared at the deficiency levels, and the same thing for terpenes. So now we have N, P, and K, nitrogen, phosphorus, and potassium, highest concentration in the deficient plant. Obviously, the grower are not going to grow hungry plants. So what we are working now in the lab on trying to trick the plants. So we give the plants optimal concentration, but give it some small treatments, trick treatments to convince the plant that the plant is under stress in order for it to produce the large amount of cannabinoids and terpenes that we can. But we continue to check. We said, this is really a general phenomenon, and we need to stress the plants for them to be deficient. So we put a few things, and this is the last result that I'm going to show you, and many other things you can find in our publications. So we checked magnesium. Magnesium also is a macronutrient, and we checked a wide range of concentration for magnesium, obviously from very deficient levels all the way to levels we expect to be toxic levels, 140. And when we look at the yield quantity, sorry, this would be quantity, we look at this as the fresh biomass. For example, of the inflorescence, these are the red dots, and we see that the highest levels are actually somewhere around 20 or 35 ppm. When we increase concentration, very little damage to the biomass. So how much flowers, how much inflorescence are being produced. And when we look at a concentration of cannabinoids, some results are here. This is not like what we saw for NP and K, for nitrogen, phosphorus, and potassium. Here we see actually the under deficiency condition of magnesium, production was very low. This is for THCA, CBDA, CBCA. These are some of the major cannabinoids. And it increases, and it's actually parallel the requirement of the plant. And this is some of the reasons because magnesium is required in the pathway for production of some of the terpenes and the cannabinoids, and for other and metabolic and other energy required processes with other ones. So that tells us that it's not stressed plants which produce high amounts of cannabinoids and terpenes. It's a deficiency or toxicity or sufficiency of specific nutrients. This is what affects this is what allows for production of more or less terpenes. And we have a lot of information now about the mechanism by which those specific elements are affecting the plant. And of course, there's a need to develop a protocol, which is what the protocols from our lab are, that gives the plant's optimal development in terms of size of the plant, size of the flowers and inflorescences, which are the yield, and so forth, but with keeping standardized level of the secondary metabolite and high level of the metabolites which are required. So sum things up. Medical value, I hope I convinced you that the medical value is affected by environmental and cultivation conditions. Mineral nutrition affects secondary metabolism in cannabis. We've seen the deficiency in nitrogen, phosphorus, and potassium stimulate production of cannabinoids and terpenes, a secondary metabolite. In terms of ammonium nitrate ratio, ammonium concentration inhibits production of secondary metabolites, so it's better if possible to go with a nitrate nutrition. And deficiency of magnesium inhibits production of secondary metabolites, so there is a correlated relationship between optimal magnesium for growth and optimal magnesium for production of cannabinoids and terpenes. And that's it. I want to say a lot of credit goes to my medical lab group. We're students. Very difficult to work with cannabis. Everything is regulated. Everything is under cameras and recorded all the time. Motion detectors, videos. So the students usually say even if they scratch their head, it goes on camera and the police sees it. But we're very grateful that while following all this bureaucracy and regulation, we can actually do the world and apply the production industry, which is booming production industry, with the information which is required. Thank you. And the funding for all this work came from the Chief Scientist Fund of the Ministry of Agriculture on Israel, which we are allowed to submit requests for funding the way that we do for tomatoes or citrus or avocado or any other legal crop.
Thank you so much, Nirit. That was amazing. I've learned a lot about medical cannabis, and I think you absolutely did convince us that the different, types of nutrition and fertilizing can really impact the medical potential for the plants. So thank you so much. I'd like to open up the conversation for some questions from the audience. So you can type your question into the chat, or we can if you would like to ask your question in person, please just indicate with your hand up and I'll invite you to ask your question. I would say that I think medical cannabis in Australia is quite new, and the people who are going into production here are probably looking at your research to really understand how we should be growing it in Australia. And the potential that it doesn't need much nutrients and fertilization is very positive, I think, for Australian agriculture with our very nutrient-poor soils here.
Yeah, Joe, I would like to mention, I mean, thank you for this comment. I mean, cannabis requires fertilization similar to what other crops do. I think there was a notion in the agronomic production system that cannabis requires a lot of fertilizers, sometimes way above what is, conventionally considered in conventional agriculture. So our studies, this is only the first step. We studied respond to individual nutrients. First of all, says, hey, be careful. You give too much, you actually damage your crop. And second of all, don't waste your money and don't waste your quality. Stick with what's required. We are not going to grow them under very low concentration, but some of the studies that we are doing now that obviously I didn't have time to talk to, it says, okay, if we want to have a very strong and large plant, let's give it a lot of fertilizers in the beginning, or let's study how much fertilizer it needs when the plant body develops, and then see how much we can restrict production during flower maturation when all the cannabis terpenes are being produced, to regulate it there to make sure that we receive large, nice producing plant, but with high quantity. So a lot of it is important to see how much the plant actually requires at different weeks of development. Also about the interaction between the nutrients. I mean, it's not the plant, obviously there are more than a dozen macro and micronutrients that the plants require. So we are working a lot on the ratio between them. And also with interaction with other things like light quality and light intensity and humidity and temperature, all those affect. So it's a whole new crop that we're closing as fast as we can the knowledge gap from.
Thank you. Erez has a question. Can I?
Yeah. Hi, Nirit. Thank you. It was a great talk and really interesting work. I just have to admit, Nirit just marked my PhD thesis a few months ago. Yeah. So thank you for this as well. I have a question. With regard to the nutrients recipe that you describe in your talk, how strain specific is it? How generic can it be? How can we have one recipe that can be applied across varied strains or like genetically remote strains, let's say? And what do you think the effect of this generic recipe will be?
Yes, thank you very much for this really important question. I have to say that most of those studies that we've done, we conducted with several varieties exactly for this reason, because we know from other crops that can be a certain variability in requirement of nutrients of different cultivars, different varieties of different cultivars. So in most of those publications that we came up with, we put results for at least 2 varieties. Overall, they seem stabler, but there is some variability. And also we know this from the cultivation greenhouses that the growers will know this variety needs more, this variety needs a little bit less. So there definitely is it's an important point, there is some cultivar variability, but the ranges don't really change that much. So for example, if we know that if we push too much nitrogen, cannabinoids and terpenes will go down, this will happen in other varieties as well. So it really showed us that I think that we need, the cannabis is just a plant, really. So, and it's agronomic plant at this point, and the range of nutrients that required are very similar to what we see in other agronomic plants. And also for some of our crops, let it be tomatoes or citrus or whatever, optimizing nutrition is expensive. This is a very expensive experiment because it requires a lot of mineral analysis and chemical analysis, even for tomatoes. So it usually is not done in agriculture for every varieties, maybe for some major varieties, but we do know to find out the ranges which are, you know, optimal. So it's a very good question, and I think that the growers eventually will, working within those ranges, will find out what's 100% suitable for their cultivars.
The fine tuning will be done by the farmers. Yes. Yeah. Okay. Thanks.
Thank you. There's another question in the chat that I'll read out from Omar, who asked, the increase in percentage of pharmaceutical compounds is per gram of tissue or the whole plant?
Great question. Because, and this is again coming back here, we have one of those deficiency plants. But who cares if this inflorescence, people call this a bud, right? It's not a bud. And it's not a flower, it's an inflorescence because it has hundreds of flowers in it. So people, who cares if this inflorescence of this bud has a very high concentration of THC, but the plant is so minute that it produces, you know, popcorn-sized, few minute inflorescence, which cannot be, you know, even cover the cost of cultivation. On the other hand, so it's all a question, as someone said, it has to be a combination between how much are we getting what's the concentration we are getting in the flower, and what are we getting per plant, or per, let's say, per unit area, per meter square of cultivation. So we in all of our papers, we always calculate concentration, and we also calculate production per plant, or sometimes per meter square. And there's no doubt, I mean, we are definitely, in this question affects a lot the recommendations that we give, because definitely we do not recommend to go with deficiency concentration as we see here. We recommend to go on a compromise, to go on somewhere where maybe where the production of the cannabinoids are not damaged totally because of high concentration, but still we're getting a good production. And as I said, we are working on protocols which will say, let's grow the plants to their best, but spray them with something or supply something to the root zone that they will sense that they are under stress. It's called to elicit stress response in order to them to actually function great, but think that they're under stress, so the production of cannabinoid and terpenes will increase. So we are fooling the plant a little bit, but it's for their good as well. So.
Fantastic. Thank you so much. I think we'll wrap it up there. We've really enjoyed your talk today, Nirit. And I put in the chat where you can learn more about Nirit's research, and also about more upcoming webinars as part of this series. Thank you all for joining today, and I hope to see you again. And thank you again, Nirit, for sharing your expertise and research with us.
Thank you, Joe, so much. And again, thank you for this really lovely initiative. And it was nice. Thanks.
Thank you so much.
Thanks, Nirit.